The overall goal of this procedure is to dissect, experimentally manipulate, and culture whole retinal explants from chicken embryos. This is accomplished by first collecting both eyes from a chicken embryo and placing them in a petri dish. The second step is to remove the scleral onlage and the pigment epithelium using fine forceps. Next, the whole retinal explant is subjected to electroporation of plasmid DNA with flat circular electrodes. The final step is to incubate the retinal explant in culture medium in a 24-well plate in an incubator. Ultimately, the whole retinal explants are fixed, cryosectioned, and analyzed by immunofluorescence microscopy to visualize the retinal neurons.
The main advantage of this technique over existing methods, like in novo electroporations, is that it is rapid, easy to perform, and ensures reproducible results. This method can help answer key questions in the field of central nervous system development, such as how neurogenesis is affected by specific chemical reagents like enzyme inhibitors or gene products that target the cell cycle.
Following incubation, place eggs at the desired embryonic age into the laminar flow hood and wipe the eggshells with 70% ethanol to avoid contamination of the embryo. Tap the side of the egg firmly against a hard surface to crack open the egg, and then place the contents into a